So we have today, Atsushi Mochizuki, he's going to be talking to us about his version of modularity in the context of chemical reaction networks.
Thank you very much. So I'm very happy to join this wonderful workshop because modularity has been very important keyword for my project. So let me first introduce my motivations. So this slide shows my interest. So I have studied dynamical behavior of the complex systems for more than 20 years. Okay, so of course we know that biological functions are emerging from dynamics of such complex systems. But of course we know that studying such dynamics of such high-dimensional systems is really difficult. And so we have our own strategy. So is taking the structural approach. So we develop mathematical theories by which we can determine important aspects of the dynamic behavior from network information only. So in other words, these theories are model-free. Now, after developing these theories, we apply these theories to actual biological systems and to derive predictions and ask our collaborator to verify whether our prediction is correct or not. Okay, so this is our approach. And today, I'm going to talk story based on the second and third theory. Okay, and I should show also one example which should encourage our interest about the modularity. So this is very famous network obtained from database. Okay, so this map shows all of the identified reactions in human cells. And of course, human cells have many biological functions, okay, emerging from this network systems, but this is a single system. So all of the reactions are connected and conform a single network. So this fact gives me large questions. So why organism can control different biological functions hopefully separately, which are emerging from single systems. And so you already agree. So modularity maybe very important concept to answer these questions. Okay, but we know actually previous concept of the modularity is of course important, but maybe less clear. And it's very difficult to define modules, and even it's very difficult to discuss the meaning of the modularity. Okay, but I hope today I hope I can provide at least one clear answer, I think. So so let me explain my modularity. So our module is substructure in reaction network, which is clearly defined from topology of the network. Okay, and as for the behavior function, our module has a property of the independent controllability from other part of the systems. So in short, substructure satisfying the first conditions, topological conditions, naturally obtain the property of the independent regulations. Okay, so the meaning of the controllability is more precisely sensitivity behaviors and bifurcation behavior. So this slide already provides my conclusions. So please keep this in your mind. Okay, so let me go to the main part of my talk. Okay, so I want to discuss about controllability, control of the biological systems, how organisms control their systems. So basically, it is by modulating enzyme activity or enzyme amount. So organisms can change expression level of the enzyme, and the change in activity or enzyme should influence amount of the substrate or product. Okay, so this sounds very natural, but this is just intuitions. And so such simple process is already interesting. I will explain. Okay. And I use a framework of the chemical reaction dynamics. And I think that I do not have to explain in detail. So we have also set of the reaction equations. And based on the reaction equations, so we construct ODE. And so change in each chemical amount is defined by the difference between total influxes and the total outfluxes. Okay. And the ODEs are so ODE is can be summarized into a simpler formula using the stoichiometric matrix. So this form or this form, okay? And yes, I called flux. The flux is also called reaction rate, and it is so common that we assume Hill functions or also Michaelis-Menten functions or mass action kinetics for each reaction rate. But in my talk, I do not assume any detailed formula for mathematical reaction rate. So we just use two trivial facts. First one is dependence on some chemicals. For example, so reaction 2 is from A to B. Okay, so reaction rate of reaction 2 should depend on the amount of the chemical A. Okay, so here we explicitly shown here. Okay. And this is first one. And the second fact is reaction parameter. So each reaction rate has a single parameter which corresponds to amount or activity of enzyme. Okay. So we consider a change in K. Okay. And so we want to discuss change in each U. Okay. And we assume a steady state. It's very common in the field of the chemical reaction systems. Okay. So steady state implies that reaction by vector is in the kernel of the stoichiometric matrix, which is expressed by the linear sum of the basis of the kernel. Okay, and which kernel is sometimes called cycles. Okay, and yes, now also we can define our mathematical problem. So we consider the response of each chemical to the change in each reaction parameter Kj under our steady state. Okay. Then this is our method, structural sensitivity analysis. So let me first claim our main message. So we can determine qualitative response of the total reaction systems. So which one? Qualitative implies which one increase, which one decrease, which one silent, can be determined from structure of the network alone. Okay. So let me explain. So I assume a steady state of the systems. So more precisely, steady state both before and after perturbation. Okay. Then change in reaction vector should be in the kernel of the stoichiometric matrix. Okay. And this equation is just also taking total derivative with respect to K. Okay. And so yes, this matrix formula is just by obtained just by linear algebra. Okay, so it's very easy actually. So this is yes, summarization into unknown part and known part. Okay, so this is what we want to determine. So response of each chemical to the perturbation of each reaction. Okay, and this part. So A matrix. This matrix is very important. So okay, so can be determined from network structure. And the right hand side is diagonal. So in conclusions, our method is just by 2 steps. So first we construct A matrix from network structure. Okay, then calculate inverse of the A matrix, and inverse of A matrix gives response of each chemicals to the perturbation of each reaction. Okay, and important point. This is an example of the A matrix corresponding to this network, and you can see 0 or non-zero entries. So actually, a pattern of 0 or non-zero are determined from network structure alone. Okay, for example, so second row corresponds to reaction 2, and reaction 2 depends only chemical A, not B, not C, not D. That's why, so we have non-zero entry here and 0, 0, 0. Okay? And the last two columns also correspond to so kernel of the circuit matrix.
Quick question. The
Yeah.
first part of the A matrix is the Jacobian, right? Or is it no? No, it's not actually. You're right. It's just the elasticity matrix.
So Jacobian is new R. Yes, later, so I will discuss.
Yeah.
So new times RJN is provides Jacobian. So this may be related to the Jacobian, but so not pure Jacobian matrix. And so in this inverse matrix, yes, of course we know some entries are non-zero and some entries are zero. And the zero or non-zero of inverse matrix should be, of course, determined from the pattern of the zero, non-zero of the original matrix. So this is a meaning that we can discuss qualitative responses, zero or non-zeros, purely from the structure of the network. And in many cases, so in case of the non-zeros, in many cases we can determine plus or minus. Okay, and I show you so very simple example. So I like this example so very much. So I always use this one. So we have also influx into the systems with chemical A, and after two steps reactions, chemical C goes out. Okay, and so we consider the perturbation into enzyme which mediate reaction 2. So you have enzyme genes which mediate reaction 2, and you increase expression level of these genes. And what will happen as a steady state? Okay, please imagine what will happen by increasing the enzyme of this reaction. Okay, I think so. Most of the people consider that. So then, flux of the reaction 2 should increase, and the downward chemical may increase. But such intuition is actually incorrect. So this is our matrix on the inverse. Okay, so perturbation into 2 correspond to the second column. And the second column implies, yes, there's no responses in downward chemicals, silent. And there's no responses in any fluxes. Only substrate chemicals should show or decrease in its concentration. Okay, but it's actually understandable. So this should be always true. So when we consider these systems, but we may assume reaction functions, okay, arbitrarily. So we should always observe these responses. So actually, soon after increasing the reaction parameter, enzyme activities, we observe increase in flux and that in downward chemicals, but they are just temporal. So they soon go back to the original level. At the same time, we should observe decrease in substrate chemical. Okay, so we can intuitively understand that. So increase in enzyme activity is compensated by decrease in substrate chemical, and there is no change in downward. Okay, and this kind of the compensations are observed in larger scale. Okay, so this is less trivial example. Now we have feedback reaction from C to A. Okay, and so chemical C has 2 output reactions, and let us consider 2 perturbations to each of output reactions from C. Okay, so I should quickly explain. So when we increase enzyme activity for reaction 4, this is a response. So this time we observe direct increase in flux of the reaction 4, and 5 to 3 show or increase in their fluxes. Okay, and the chemical D, A, B should increase, but C is silent. Okay, and when we increase reaction parameter for reaction 6, this is a response. Okay, now 6 is silent, and we rather observe a decrease in flux of reaction 4, 5 to 3. And so chemicals D, A, B, C show or decrease in their concentrations. Okay, so between left and right, similar part show responses. Directions may be different, but please be careful. In left, C is silent. Right, C decreases. Okay, So we gave locally symmetric perturbations, but we obtained very different responses. Okay. So yes, in all of the cases, we observed confined responses, and the patterns may be counterintuitive. Okay. So actually, so these patterns, all of these patterns can be understood by our theorem, law of localizations we named. But today, so okay, yeah.
Isn't this explicable just because
Sorry,
increase in reaction decreases the substrate? Isn't that just sorry.
Sorry.
Yeah, you're saying it's counterintuitive. Isn't it just explicable by the fact that increasing the downward reaction decreases the upstream substrate? Or is it more complicated than that intuitively?
So maybe you can give us intuitions.
No,
but
no, I'm just trying to understand.
Hmm.
Like for instance, in your very first example. Sorry. Oh,
Maybe so these responses are yes, this response is here maybe closely related to the first responses.
Yeah, so the first
It's on.
the first one is actually not too difficult to understand. The reason why that's happening is because the initial reaction is fixed.
Yeah.
And so all subsequent reactions have to maintain that same pace. And so you can logically deduce, for example, the last circle has to remain constant because you haven't changed the enzyme level on the last reaction. And the only way to maintain the same flux that's been fed in is for that second species not to change.
Yeah. So,
That's it.
So so we have only also one influxes, which is constant. So the system is linear. So all of the flux should not change. It cannot change. But in these cases, so we have two kernel bases. One is running from top to end. So this flux should not change. But so we have another kernel here, and this should have yes, freedom. But yes, so we can understand flux changes are confined. So this cycle is maybe understandable, but response of these chemicals may be difficult to understand.
Second one, the last circle also won't change because you haven't changed the outflow. And because you're feeding in a fixed flux, which is the same as the flux that's coming out, the last circle cannot change.
I mean, first kernel is running. So
Yeah, but the mass coming in has to be the same as the mass coming out.
Right.
So since you haven't changed the last reaction, in order for the same amount of mass to leave, that species has to remain the same.
Yes, this one cannot.
That one.
Yeah, this one cannot change.
Yeah, that was.
But this one receives perturbations, and to compensate given perturbations, this one change.
Yeah.
So that is induced or side effect of the change in this cycle.
Yeah, that one is more interesting.
Yeah, this is one way to give us interpretations. But also we have series of the very interesting patterns. So when we see this kind of confinement in multiple scales, so much larger scales, we may see also confined responses. And so the region of the confinement becomes larger and larger. Then our intuitions concerning our intuition should be difficult and more difficult. Okay, but yes, this one, yes, generally, universally explains the reason of the confined responses. Okay, and today, I emphasize the name of the substructure, which correspond to module, I think. Okay, so yes, we focus on the substructure of the reaction network. And if substructure satisfies these conditions, the substructure naturally obtains special properties. As I will explain one by one. So focusing on substructure implies selecting subset of the chemicals and subset of the reactions. And the first condition implies that after selecting chemical subsets, all of the emanating arrow from selected chemical should be selected into reaction subset. Okay? The second one is interesting. So second one implies that. So let us count the number of the element in the selected structure. Number of the chemicals and the number of the reactions and the number of the cycles and number of the conserved quantities. So if these four integers satisfies these equations, then the substructure is buffering structure. And what is buffering structure? So any perturbations inside, any perturbations into parameter inside a buffering structure influences only inside chemical concentrations or inside the reaction fluxes, and there should be no response outside at steady state. Okay? And so this is also analog of the Euler characteristics. Okay? And actually, proof is very simple, very easy. So buffering structure corresponds to square non-zero block in A matrix. So this part should be a 0 or from the condition 1. And horizontal and vertical length should be equal from the second condition. Okay? And please recall undergraduate math. So if a regular matrix possesses this structure, square, then inverse matrix should possess the same structure. Okay? And the 0 block here in inverse matrix implies response of outside should be always 0 to the perturbation into inside. So actually, our law is very natural. Okay? Yes.
You've written it. You have the hypotheses and then the conclusion about the book.
My understanding is you've got the hypothesis 1 and 2 implies the perturbation inside the buffering structure.
No. These two just correspond to the patterns in A matrix. So these two are not hypotheses. So this is a description. So about the patterns of non-zero entries in A matrix.
Yes.
Yeah.
So
Substructure satisfying these two conditions construct square non-zero block in A matrix. Okay. So, but satisfying this implies these patterns. So, which implies response. Non-zero responses outside.
Okay, so that's the implication. Now, does it go the other way? If I have a substructure and any perturbation you cannot find a perturbation that impacts outside of that structure, must 1 and 2 be satisfied?
Very good. Yeah, very good comment. So you are asking about the inverse.
Yes.
Yes. Inverse is also true.
Okay.
Yes. Okay. So I'm very happy. So I should get it. Yeah. So let me explain. Yes. So law of localization claims that. So if local structure satisfies topological conditions, then we should observe confined responses. So recently, we could obtain the inverse theorem. So if we observe confined response also in sensitivity meanings, okay. So I mean, so if there exists any chemicals showing 0 responses, then there should exist corresponding buffering structure. So yes, so 1-to-1 relation between structure and behavior. Okay.
One more question about your block matrix. Doesn't that depend on the ordering of your rows and columns? Doesn't the block matrix? I mean, I could reorder the rows and columns, and I won't see the block matrix. So there's a particular ordering you're doing here.
So we have to select. So when we also collect, so after selecting M and E, so we collect columns and rows, so which associate to selected ones. Then if we have squares, so we should observe the confined responses.
Okay.
So we need to, so actually, so we need to correct. So, yes, rearrangement of the column and the rows. Okay, so this is just also, yes, look back our examples. So in this case, so yes, minimum buffering structure, one chemical, one reactions, and also no cycles, no concept quantity satisfies our conditions. And so in example 2, left case, this is buffering structure, 3 chemicals, 4 reactions, and also 1 cycle. And our cycle is defined in so reaction space. So this is cycle, okay, satisfies our conditions. And in right case, this is buffering structure, okay, 4 chemicals and 5 reactions and 1 cycles. Yes,
Just so that I understand the statement. So we have to do this perturbation everywhere, right? So we have to do this perturbation everywhere and check that this is 0.
So when we consider any possible perturbations, so we should search minimum buffering structure, including the part of the reactions. Okay. Then the buffering structure specify the range of the response.
Okay. And do we also know the number of conserved quantities, or like how do we know the number of conserved quantities? Do we have to simulate the perturbation and check?
Actually, so number of the independent concept quantity can be determined from the co-kernel of the stoichiometric matrix.
Okay.
So actually, so what we call number of the cycle is dimension of the kernel space. Which has the kernels which has support in the selected reactions. So this corresponds to the dimension of the co-kernel of the stoichiometric matrix. Okay. Okay, so yes, so all of the cases can be understood by buffering structures. This is most important message. Take home message, kind of take home message. So actually, modularity. Modularity in chemical reaction network. So naturally emerge from substructure when the substructure satisfies the topological conditions. So let me express differently. In evolutionary history, substructure may satisfy 2 conditions just by chance. Okay, but once substructure satisfies conditions, the substructure obtains biological functions as regulatory module. Then I think the structure might be conserved in evolutionary history. Yeah.
Sorry, just another observation. Go back to the previous slide. Yeah. Yes. This goes back to a theme, I'm not sure, by Dr. Baliga's. It seems examples, in example 2, the 2 modules that you've defined are different and depend on where the perturbation is, right? Structurally, they're the same, right?
Sorry,
in example 2, you have 2 versions of it.
2 versions? Yes. So let us recall the discussion with you. So we should consider the minimum buffering structure, including the part of the reactions. So in this, when we consider perturbation into these reactions, this is the smallest buffering structure, including these reactions. Okay, so maybe I should mention. So there exists buffering structure here, corresponding to this one. There another smallest buffering structure here, okay, and smallest buffering structure here. And so by adding these reactions, this is the smallest buffering structure. Okay, so when we consider the perturbation into these reactions, this is the smallest buffering structure.
Right. All I'm saying is that the modules you define then become defined by where the perturbations are. So the modules are not predefined. That's all I'm saying.
I do not understand.
Maybe we can talk.
Heading of modules. We can keep going.
So yes, maybe I should quick. Yes, this slide is maybe important to increase. Yes, let me quickly explain. The same things also may happen when we consider the perturbation sensitivity in regulatory systems. So when we consider the response of the regulatory systems under the growth rate perturbations, we consider the inverse of the Jacobian matrix. Okay, and so if Jacobian matrix has square non-zero block, then of course we should observe confined responses. I think this idea may correspond to the suggestion by Reinhardt. He mentioned about the strongly connected substructure. So this is correspond to, I think. But in case of the reaction network, the responsible matrix possesses complex structure. So the meaning of the columns and rows are different, including multiple different entries. Okay, so the index, the square block need also special definitions, also which correspond to the buffering structure. Okay, this is our understanding. Okay, and so this is example of observed in example of the buffering structure observed in real biological systems. So it is cell cycle systems. So cell cycle is actually not periodic oscillations. The important biological function of cell cycle is rather stopping the process. So between one cell divisions and the next cell divisions, there are 4 different phases. So in case of the yeast, and transition between phase, especially from G1 and S and G2 and M are strictly controlled. So a cell can invade into S phase only when protein complex, so here we call G1S complex, increases. And a cell can invade into M phase, so only when another protein complex, G2M complex, increases. Okay, and these two are different species of the protein complexes, but they share a common protein, so which is called CDK. CDK is a very famous protein. And so it is well known that amount of the CDK is almost kept constant in all of the phases of the cell cycles. Okay? Then, yes, this is a reaction network. And so here we have G1S complex and G2M complex. And so there are 6 species of the protein complexes which include CDK. So this implies that. So there are 6 species. Total amount of which should be kept constant. Okay? Then people consider intuitively that. So the amount of this one and this one may be negatively correlated. Increase in G1S complex may directly imply decrease in G2M. Okay? This is just intuitions, but maybe interesting. So let us consider whether this intuition is correct. So we just apply our structural sensitivity analysis, and so we usually examine vertically this matrix. But so let us see horizontally. So this is G1S complex, and this is G2M complex. And we see which parameter influences amount of each of them. Okay? And I should plot on the graph. Okay. There are only 2 reaction parameters which influence amount of G1S, and there are 6 reaction parameters which influence amount of the G2M. And they are disjoint. There is no overlap. So no overlap implies this intuition is incorrect. Okay? So yes, it rather implies. So these 2 are independently controlled. So this is just a demonstration of the independent control. So we calculate the dynamics of these systems, and while calculating dynamics, I change synthesis rate parameter for SIG2, which is part of this complex, and we see increase in G1S complex. But sorry, so this is steady state plot. Okay, but so another G2M complex is silent at this time. Okay. And then we increase synthesis rate of CDC13, which is part of G2M, and we see an increase in G2M, but also G1S is silent. Okay, so this is the meaning of the independent control. And actually, this embedded independent control is realized by 2 buffering structures. So this is the largest buffering structure, including G1S, which does not include G2M. So any parameters which may influence G1S complex in these systems never influence G2N. Okay? And similarly, this is the largest buffering structure including G2N, okay? Which does not include G1S. So any reaction parameters which may influence G2M in this system never influence G1S. Okay? So very beautiful mutually exclusive regulations by two buffering structures. And so now, we are also collaborating with experimental biologists to ask the verifications. And the verification experiment is very interesting, but so I show just part. So at least part is, yes, examine. So they increase the synthesis rate of CDC13. So they increase very much. But so, yes, we actually observe increase in G2M, but so G1S never changes. So clearly showing existence of the blue buffering structure. Okay. Okay. So this is a summary of first part. So yes, so we can determine qualitative response of reaction system to perturbation changes from network structure alone. And under this, process sensitivities. So yes, buffering structure takes very important role. So this buffering structure create independent regulations from other part of the total systems. Okay.
We have 2 minutes.
2 minutes.
Yeah.
Okay, just mention. Okay. So actually, yes. So buffering structure govern another important property. So bifurcations. Okay. And so I should not. So essence is that. So yes. So this part is so coming from the our finding that Jacobian matrix and A matrix possesses the same informations. So yes, we usually also identify steady state bifurcations by determinant Jacobian equals 0. But also we can do the same thing using determinant A equals 0. Okay? So this implies that. So there are many advantages. First one is we can discuss condition of the bifurcations from network structure because A matrix directly reflect structure of the network. One non-zero entries correspond to one arrow in chemical reaction network. And the second merit is, of course, coming from buffering structure. So if A matrix possesses buffering structure, then determinant A can be factorized like this, corresponding to buffering structure and corresponding to outside of the buffering structure. So this implies that. So bifurcation condition can be decomposed into two parts. Either buffering structure satisfies bifurcation conditions, or outside of buffering structure satisfies conditions. Okay, so in this way, so we can decompose bifurcation condition of the total large system into smaller part based on the buffering structure and examine the possibility of the occurrence of the bifurcation for each of the decomposed structure. Okay, so I will skip the major part, and so you can imagine what we did. So from the figure, yeah, so we can do. Yes, yeah. So yes, so we predict bifurcation behavior from the network topology alone, and also confirm by assuming the reaction functions, and so they can be they follow our prediction completely, and also we examine very large systems. Okay, so many buffering structure included in systems, so we can decompose into very small substructures. Okay, so we see many regulatory modules and a hierarchy in regulatory modules, and we also see there are bifurcations conditions. Yes, in this case, after decomposing into 17 terms, only one can possess 0 values. The other terms are always positive or negative. So condition is very simple. So we can focus only one one term. Okay, and so numerical simulation follow our predictions. Okay, So all of these things can be determined from network topology before assuming all functions. Okay? So this is my viewpoint. I think in real biological systems, there must be many buffering structures, and they realize independent regulations between different biological functions. This is just also, yes, image, but also, yeah, I think so. Our conjecture would be true, would be correct. Okay, so this is the summary of the talk. Okay, so I should stop. So thank you very much for your attention and very